Hello, everyone, and welcome to another eye opening episode of Lab Talks with EPTOS, your window into the people and ideas shaping the future of science and innovation. I'm your host, Dr. Purnima, and today we are diving into the fascinating world of organoid research, which is often also called as creating the mini organs in a dish. Imagine being able to replicate the tiny versions of human organs in a lab to study the diseases, discover the drugs, and even personalize your treatments. That's the promise of this field, and it's changing the way we think about medicine. So I'm thrilled to be joined by Dr. Rishika, a scientist whose research focused on the organotypic skin models and the organoid cultures. What makes her work especially exciting is that it connects the fundamentals of cell biology with the real world applications in drug discovery, disease modeling, and the alternatives to the animal testing. She has been closely following how this field is evolving globally, and today she's here to help us break it down into the simple, relatable terms. So, Dr. Rishika, welcome to the Lab Talks. It's a pleasure to have you here.
Thank you so much for having me here. I'm very excited to be here and looking forward to talk about organoids.
Great. So let's start with the basics. Basically, organoids, often described as mini organs in a dish. For someone who have never heard of them, how do you explain what the organoid research is and why it's such a big deal?
Yes, absolutely. So traditionally, we scientists have grown cells in the lab to uh, to study and represent what we see in tissues. However, when we culture cells, we grow them in a petri dish. So think of that as a flat plate, and we grow the cells on top of that. And so these cells kind of grow out as a jigsaw puzzle. And while this
Yeah.
jigsaw puzzle gives a good two-dimensional representation of the organ, our organs are not two-dimensional, they are three-dimensional. And so when we grow organoids, what we are doing there is we are giving the cells a third dimension. And so instead of growing out as a puzzle, they grow out as a set of Lego blocks growing on top of one another. And so this is a more truer representation of a tissue or a part of a tissue. And that's what we call as organoids.
So basically, I love your Lego versus puzzle analogy. It makes it so easy to visualize, and it also highlights why this 3D culture matters. So, building on that, what makes the organoids or 3D culture models more powerful as compared to those traditional 2D cultures?
Yeah, so in two-dimensional cultures, the cells they lack some of the structural and functional features that you see in the organs or tissues. But when you give them this third dimension to grow, the cells have this enhanced volumetric space to grow, and they can basically express proteins that they don't grow when they are grown in 2D cultures. So, for instance, when we grow liver cells, hepatocytes in culture, they might lack some of the functionality that you would expect from them. Or similarly, when we grow skin organoids, uh, our epidermal cells, keratinocytes, they don't express a lot of the proteins that they actually express in the body. And so this really gives the organoids the added advantage compared to two-dimensional cultures.
So, yeah, great to know about this 2D and 3D cultures. Then, definitely, this must bring with some future opportunities and the different healthcare developments. So, how do you see these being applied in the healthcare drug development, and where is the immediate impact of this all?
Yes, so where we are seeing organoids being used is to study disease, test drugs, as well as develop new therapies. Plus, in uh, US as well as EU are coming up with new regulations to reduce animal experimentation. And so, if we reduce animal experimentation, we need new technologies and new methods to study these compounds. And so organoids have emerged as these new alternative methodologies or NAMs to do these studies. Then, in addition to this, organoids are also really expanding the personalized medicine space. because you can isolate a few cells from a patient, uh, expand them in the lab, culture them as organoids, and then test the drugs. So let's say the doctor or the physician could prescribe 5 drugs, but they don't really know which one the best the patient would respond to. And so if you could test this in the lab and then prescribe the best one to the patient. So I think that's something super cool about organoids. And then also finally with AI speeding up drug discovery, and like hundreds of thousands of new molecules are being discovered. And these have to be tested in the lab before they can move into the clinic. And so organoids also really makes this very high throughput, as well as gives more confidence in the data compared to two-dimensional cell cultures.
Yeah, really, it sounds interesting because, like, emerging as a natural alternative to opening the doors for the personalized medicine, and then definitely with AI speeding up whole of this drug discovery, definitely it's quite interesting. But I imagine it's not always a smooth sailing. So, what do you think? What are the some key challenges that the institutions face while advancing this whole of the organoid research? Because this field continues to grow worldwide.
Yes, so organoids are a relatively new field of research. They've only been around, I would say, for the last 30, 35 years or so. And only recently has the research really grown a lot into them. So the first challenge that comes up is standardization of how we do this. So labs usually have a very set protocol for two-dimensional cultures. You look up a protocol for a certain cell line, and you get the protocol on the internet, and I would say 90% of the labs follow the same protocol. This is not really true for organoids, because labs have kind of developed their own protocols, so there's a lot of variability, and so there needs to be some kind of a regulatory body that can standardize these protocols. Then in addition to this, organoids also have some variability, especially if you're focusing on, isolating, generating organoids from patient-derived cells. Uh, this variability just exists because, as humans, we are variable from each other. And so while it is a benefit, it is also a challenge because you have to standardize the variability, which is, uh, kind of a difficult thing to do. And then finally, scaling up organoids is it's a costly affair, and so we need, of course, more research funding into it. Plus, in addition to that, tools that are equipped to grow and scale organoids. Like we have them for the cells, but we now need to develop these, lab kits and equipment that can allow us to scale organoids.
Definitely, that sounds futuristic. Because scaling up, costs, collaborations, technological advancements. These are definitely the points which truly drive this evolution of healthcare. So, finally, to wrap up, let me ask you this: looking ahead, how do you envision the organoid research shaping the future of medicine over the coming next, let's say, 5 to 10 years?
Yeah, I think it's a technology that will become almost as commonplace as your two-dimensional cultures in the lab, because more and more labs are adopting it. And then in addition to it, it'll basically lead to it becoming, central to how we study the human biology. Um, I also see it, helping us in reducing dependence on animal testing, and to model diseases better in the lab, to understand the disease better, and then hence, develop better drugs for it. And then finally, I think organoids in combination with AI will be a super useful tool, that really has the potential to change the landscape of healthcare research.
Definitely, that being coming up as an integral tool for both research and clinical practices, that's really inspiring and really shows that how science can bring us closer to the more ethical, efficient, and more of patient-centered healthcare.
Yeah, absolutely. It's been really lovely speaking to you about organoids. I've spent, the last 6 years of my life doing research on organoids, and I'm very excited to see how the field grows and where it leads us. And this is such a collaborative space, but I think it also has the potential to be even more collaborative, between academia, industry, startups. And this has also kind of led me to realize the importance of how uh, useful skill it is, to be able to communicate the science that you're doing and talk about its impact. And this has led me to, do what I do at Biomed Sapiens, where I help students and startups learn how to communicate their science more effectively. So super excited to see where the organoids take us.
So thank you so much. It was really inspiring to have this all information coming from your side. Thank you so much for joining us today and making this complex topic so approachable. We truly appreciate you sharing your insights with us today. And definitely to our listeners, whether you are in a lab coat or just curious about how the science works behind the scenes, we hope you enjoyed today's deep dive into the organoids. We will be back with more conversations with people who are shaping the tomorrow's science. Don't forget to follow, share, and send on your questions for the next time. And until then, stay curious and keep advancing science with quality. Thank you.
Abdos Life Sciences. Advancing science with quality.